Naked?
Nope.
No.
Naked viruses don't have an envelope. They don't have any, all they have is a nucleocapsid.
A capsid. Okay.
That's it. Just a protein coat, but it's pretty indestructible. So here's some of the questions I'm going to ask you. Where does the envelope come from on an envelope virus? The plasma membrane of the host cell. It buds through it. And what does that envelope do for the virus? Allows it to attach to the same kind of host cell. And if you destroy the envelope, what happens to that virus? It's destroyed as well. All right? What about a virus that doesn't have an envelope? What's it called? Naked. And naked viruses, are they easily destroyed?
No.
No. How about enveloped?
Yes.
Name some things that will destroy enveloped viruses.
Alcohol.
Detergents, alcohol, disinfectants, antiseptics, phenol, all of those things like Lysol, anything with a detergent in it. Now remember, there's a difference between a detergent and a soap. All a soap is an alkali plus an emulsifying agent. So you have a very high pH, that's what alkali have, and what it does is it lifts oil and the dirt dissolved in it off your hands, and physically with the foam washes it down the drain. A detergent does the same thing, plus it inactivates enveloped viruses and bacteria. But most people have watched that television program that's been on since 1940, that television commercial where Madge is soaking in palmolive. And they said, oh, she's going for her manicure, and she says, oh, my hands have been so rough. And she says, yes, you should change to a better dishwashing detergent. Something like palmolive that's kind to your hands and your skin. And she said, well, I'm worried about using something like that. She said, well, you're soaking in it. And she goes, oh! And then says, well, it's fine because it's okay. Well, remember, detergents have a reputation for drying and making chapped, cracked hands, which they do. But soaps don't kill chemically, only physically. So you want, but could Dial sell Dial antibacterial detergent to you to wash your hands with? You wouldn't do it. But what did they change it to? Dial antibacterial soap. Is it really a soap? No, it's a detergent, but they couldn't market it as a detergent because people thought, oh, it'll dry out and crack my hands. So they sell it as Dial antibacterial soap, but it's not a soap. Now, one other thing, remember, anybody, most of you women in here have knowledge about this, most of the guys don't, but I'm sure you know that if you're taking care of your beautiful hair, you don't use Pert. Does anyone know what Pert is? Pert is this product that they sell that says it is a shampoo and a conditioner in one. You can shampoo and condition at the same time. You cannot. Conditioners are oils. Shampoos are detergents. What do detergents do? They remove oils. So you cannot add an oil to a detergent. If you do, you've done nothing. So when you go and buy something to wash your hands with, I wish we used to have some here, I guess we don't. You want to buy an antibacterial soap, but you don't want to buy one containing lanolin or skin softeners because it's an oil. Oils hide bacteria and dirt. So it's sort of like this. You can't jump on top of the car and go to school. You got to get in, you got to go through the process. And so you wash your hands very well with a detergent, and then you go get a good hand cream to moisturize them and keep them from cracking. You don't try to do it all at once. I know they try to sell you the product, but it won't work. In fact, it's more hurtful because it traps dirt and bacteria in the oil droplets. So remember, you've got to wash first, then moisturize. Never try to do it at once. So emerging viruses are viruses we've never seen before because we didn't live there. You know what they used to call what's that area around San Bernardino called what do they call that? The something inland empire. You know, they called along here the Gold Coast. They call that the Inland Empire, which is marketed. But what did we used to call the Inland Empire 20 years ago? Desert. But you don't want to say, let's move to the desert. Move to the Inland Empire. Now, so what was the Inland Empire? Desert. How many people lived there? 5 per every 10 square miles. How many people live there now? 500 per square mile. So we had a disease that didn't exist, or we didn't know about it. It existed for 5,000 years that we know of because the Navajo talk about it. And it's called, now we call it Southwestern Pulmonary Honton, which I never spell right, I hope it's right, virus syndrome. And it's carried by the California deer mouse. The normal brown mouse that you see running around here is a deer mouse. It has ears that look just like a deer. That's why they call it the deer mouse. And it has this virus living inside of it happily. They coexist. They love each other. They're symbiotic. The virus doesn't hurt the mouse, and the mouse doesn't attack the virus. They live together in harmony and love. And whenever the mouse poops or pees, they pee out, just like we poop or pee out anything we're infected with. They poop or pee out this virus. But when humans breathe dust that this virus has peed on, that these mice have peed on, you get Southwestern Pulmonary Hantavirus Syndrome, and it will kill you in 4 days.
Wow.
And it kills you by your own immune system killing you. You kill yourself. Your immune system kills you. So here's what happens. We didn't see that virus because people lived there 5 for every 10 miles, 10 square miles. How often would they come across mouse pee? Hardly ever. Then we moved in and we planted these lovely lush lawns that we irrigate, which produce seed, which is what mice eat. And they move in and they eat and they live with us and they live in our garages. And then on the weekends we say, oh my God, the garage is just such a mess. So all the kids and I are going to get out there and we're going to move everything out of the garage and we're going to dust it and then we're going to move it all back in and organize it. And you breathe the dust. And by the afternoon, by the evening, you have trouble breathing. Your lungs are filling up with fluid. They take you to the hospital. The hospital doesn't think about this disease because it's used to be so rare. And remember, your doctors are trained, what, 10, 15 years ago. This disease wasn't even seen until 1997. So they don't think about it because it has all the symptoms of viral pneumonia. But it progresses so fast that you will be dead by day after tomorrow. Now what they're supposed to do is put you on a respirator and give you steroids to cut off your immune system. But that's not what they normally will do because they haven't isolated and identified it, and they don't want to cut off your immune system if it's a bacterial or another kind. So what happens is the virus that you breathe in lodges in the cells of your alveoli, like the little capillaries lining the alveoli that absorb oxygen. in your bloodstream are what we call cytotoxic T cells, killer T cells. And these killer T cells will attack any host cell that's infected with a virus. Now, if your killer C cells don't attack your alveoli lining cells, fine. The virus will reproduce, it will leave, no one gets sick. But what happens is your killer T cells will destroy the lining of the alveoli. Tissue fluid from the bloodstream will leak and fill the alveoli. Your lungs will fill up with fluid and you'll die. So your own immune system kills you. And this virus is what we call an emerging virus. Why? We changed the environment. We moved in by the hundreds of thousands. We then watered everything, made lots of food for them. They moved in with more food. There are going to be more mice, more people come in contact with urine that has soaked dust. Who dies? People who clean out their garage or their little shed where they keep their tools in their yard, or sweep out a cabin up at Big Bear. People die. They die from breathing the dust. So what can you do? Well, one, you need to be aware of the symptoms, and it's very rapid. Tissue fluid filling the lungs. So if you find someone that cleaned out their garage and by the afternoon they're having trouble breathing and they feel very bad, get them to the hospital emergency room and suggest Southwestern Pulmonary Syndrome, and they can treat them and they will live. But most hospitals won't, because they're going to eliminate all the other things before they give you steroids and put you on a respirator. They'll probably put you on a respirator but not cut off the immune system. So this virus is an emerging one because we changed the environment and we are in huge numbers where we didn't used to be, and we provided so much food and stuff that they could reproduce in huge numbers. What can you do? Well, of course, you would like to wear a well-fitted mask, and that's kind of hard to do. If you go and get one of these masks from not a hospital mask, but the ones with the little filters on it. And also, if you don't sweep, you do what the city tells you not to do, which is use a hose to wash out your garage rather than sweeping it. It's much safer. So it's an emerging. An emergency means it's been there for thousands of years. Most people think these viruses are wild mutants. There aren't. They're not wild mutants. They've been there forever. We just haven't been there in huge numbers, and we've altered the environment so that now it encourages them. And reemergences, that's the viruses that are back. Anyone have any problems with anything on this? Okay, viral evolution, we believe that since lots of people think they came first because they're simpler. But remember, they all need a host. So they probably either evolved together or evolved after host cells. And then some people believe, no one knows, these are just ideas, that viruses are reverse evolution. They are cells that have lost all their other parts except for the nucleus. So there's all kinds of ideas. Some people think they came from plasmids. So interesting ideas, but what do we know for sure? Nothing. Okay, remember bacteriophages? This is the way they look. These tail fibers is how they attach to species, and they are all DNA. They cannot, it says here that they can be single-stranded RNA, but so far as we know there's only been one or two, and that's not super confirmed. So we're going to go with they are overwhelmingly DNA, just like the viroids and the cytolides are overwhelmingly RNA. We do not give them names anymore. We just say what the host is and give them a number. You'll see them as E. coli T-Pod 40. Okay, what do you need to know about this? Every different kind of virus reproduces slightly different. So I'm not going to ask you to know RNA animal viruses or DNA animal viruses or RNA plant viruses, RNA animal viruses or bacterial viruses. Let's just know the steps, because there's some slight differences in each one, but the steps are always the same. When a virus sticks to its host cell, specifically by these receptors, it's called adsorption, not absorption, but adsorption. Then it's going to inject its DNA or RNA into the host cell. Now it can inject it, or it can actually bud into it or fuse into it, but no matter what, it gets its genetic material into the host cell, and that's called penetration. On those bacteriophages, they release a little enzyme that cuts a hole. All right, next, the DNA or RNA of the virus takes over the host cell and tells it, make virus parts. And that's called synthesis. Then the parts are assembled into whole virions, and that's called maturation. And then they either bud out or explode the cell, and that's called release. So at least know the steps and what generally happens in each one. There is a word that I wanted to remember. Let's see if I can think about what it was. There is no such thing as a little virus or a baby virus or an immature virus. They don't mature, they don't change, they don't do anything. They're either complete or incomplete. Complete, they are capable of causing infection. Incomplete, they're not fully matured or assembled. So what do you call one that is fully assembled and capable of infection? Virion. So you will often see, how many virions does it take to cause HIV infection? Well, it's 250,000 virions per milliliter of blood. Each one, remember, has a minimum infectious dose. So, adsorption is attachment, penetration is how the DNA or RNA genetic material gets in the host. Synthesis is when it takes over and makes particles, makes pieces of viruses. Then maturation is when they're put together to the intact virion. And release is budding or exploding out.
So a virion is a piece of a virus?
Nope, it's a whole virus that's ready to infect. There is no such thing as a little virus, a baby virus, or an immature virus.
Well, then what's
There's
the
either
difference?
assembled or not assembled. And if they're assembled, they're virion. Assembled and capable of infection.
What's the difference between a virion and a virus?
One particle that's capable of infection is a virion, and a virus is the group. This is just talking about virus nomenclature. Remember all those rules about genus and species? Forget them. Don't apply to anything smaller than a cell. How do we name viruses? Well, bacteriophage were discovered before we set up any nomenclature, so it's all bizarre. Generally speaking now, they're named by what they infect with some number. Let's talk about the rest of the viruses. And they are separated into two different groups. If it has DNA inside or RNA. Then, how is the DNA or RNA arranged? Does it have a capsid or not? Does it have an envelope or not? And then finally, by the disease it causes. So, remember, RNA or DNA, arrangement of the genome, shape of the capsid, whether or not it has an envelope, and the disease it caused is how we organize them. And here is the organization. This is RNA viruses. What do you have to know? Not much, just a few little things on this, like what's the difference between positive and negative? What do these things stand for? This stands for single-stranded. This stands for little pieces of single-stranded. Segmented, single-stranded. Segmented, double-stranded. Single-stranded, two-stranded. All right, what's positive or negative mean? Anybody have an idea? No? What? No? Can it act as messenger RNA? If it's an RNA virus and it can actually code for a protein, it's positive. If it has to be converted to something else first, converted to positive, and then to DNA, or converted to DNA directly from negative, then it is called negative. So remember, this is RNA. If it can be translated directly, then it's positive. And this one is the one that causes AIDS and many of the cancers. It's the retroviruses. It causes leukemia and AIDS and tumors and other kinds of cancers. And what's interesting about it is there are two pieces of RNA inside the capsid. Either one is capable of causing infection, which makes it almost impossible to destroy, because if you destroy one, you got the backup. So that's really evil. I mean, here it has two ways of infecting you in one particle. Two pieces. Both capable. All right, oh, and look up underneath where it says classification of viruses, RNA viruses. What is more dangerous, DNA viruses or RNA? RNA. Why? Because remember, they have to go that extra step. They've got to convert RNA to DNA before they can put their DNA in your DNA. So they have to go an extra step. That means more mutation equal more danger. And then secondly, and most importantly, there are no repair mechanisms for fixing mutations of RNA. Remember, there are 3 repair mechanisms for DNA, but there are none for RNA. So if I ask you, and I will on the test, I'll say, have some multiple choice questions that say, why are RNA viruses more dangerous than DNA? Answer, number 1, an extra step equal more chances for mutation, which makes them more likely to cause problems. And number 2, the big one, there are no repair mechanisms for damage to RNA. Okay, so moving on to DNA. Oh, let's just look at the diseases. Polio, the common cold, hepatitis A, all class 1 RNA viruses. Equine encephalitis, rubella, yellow fever, class 1B. Measles, mumps, and rabies, class 2. Influenza, Ebola, southwestern pulmonary syndrome, class 3. Class 4 are these rotaviruses and reoviruses. Those are those diarrhea syndromes that babies get that spread through and can kill them real quickly. So that's that gastrointestinal and that virus. You know how every once in a while, usually during the winter, people will get this thing that gives them a headache and they sneeze like they've got a cold, but 3 days later they're fine. That's a reovirus. Very infectious, and it's naked, so there's hardly any way, even washing your hands is not going to stop the spread of it. And how do you tell it from a cold? Same symptoms at the beginning. You get over it in 3 days. A cold is always 14. And then, of course, the last one, class 5, that's AIDS and leukemia.
Does the shape have to do with anything?
Nope. It just tells you whether it's, has that weird shape, or if it's shaped like a stop sign, or just helps you if you're taking a picture of it with an electron microscope, you can tell a little bit about it. DNA viruses, again, lots of respiratory infections that come around, and again, they're naked, which means that you're not going to be able to wash them off. You can physically lift them off. Our herpes, our chickenpox, our shingles, EBV, Epstein-Barr, mononucleosis are all DNA. Smallpox, cowpox, monkeypox, warts, cervical cancer, parvovirus, roseola, and hepatitis B. Those are all DNA viruses. This you don't need to know. How do we grow them? Well, we grow bacterial viruses on bacteria. We actually take a plate with agar in it, and we cover the plate solidly with the bacterial host, like E. coli. Then we go order an E. coli phage and put it on here, and if it eats a hole in the lawn, the bacterial lawn, if it eats, these are called plaques. And you can actually figure out how many viruses it would take to make that many plaques. Plaques are holes in the lawn of the host bacteria. So that's the way we grow bacteriophage. In the old days, back when we were rich and had much longer term, we would go to the sewage treatment plant and collect sewage and isolate E. coli phage from it. And we'd do an experiment where we would grow E. coli on a plate, and then we'd take liquid that we collected from the sewage treatment plant, because where would you think a lot of bacterial viruses would be? E. coli viruses would be in an E. coli system, which is a sewage treatment plant. And we would then get plaques where they had killed the E. coli on our plate from the liquid in a sewage treatment plant. So that was bacterial viruses. All right, how do we grow the viruses on plants, or like viruses that we want to infect you, or we want to do experiments on? We do it usually in what we call monolayers. There are two ways to grow human viruses. If you're going to grow blood cell viruses, you would do it in a chemostat. But if you're going to grow like viruses like herpes that grow on epithelial cells, you get a plastic flask that looks like this, and you grow them on a single layer of human cells at the bottom called a monolayer. And what you do is, you can get this monolayer from wherever you get, whatever the host cell is. Like if I wanted to get a monolayer from her, I just take a pair of tweezers and go over there and I'll just gouge a chunk out of her. Put it in that plastic flask, add a little enzyme that would break apart the cells, and then add what feeds your cells? We talked about this before. What feeds every cell in your body? Circulating in your blood, what's the majority of your blood made of? Blood serum. So we would add fetal calf serum, because that would be what we feed it. We would add a phenyl red phosphate buffer to keep the pH constant. Why phenyl red? Because we would want to know when your human cells had eaten all the food and produced a lot of acid poop. So phenyl red phosphate buffer. And we don't want bacteria to fall in this and cause problems, so we would add antibiotics. So to grow viruses, we would first take a chunk of the host cell, then we would add an enzyme that breaks them apart, and then we would add fetal calf serum, phosphate buffer, antibiotics, and a few vitamins and minerals. Lay it on its side, and the human cells will attach on the bottom, and when they eat all the food in the fetal calf serum, it'll turn yellow. And you pour it off, and you pour more back on, and the human cells are sticking to the plastic. And they're dividing one by one. And as they divide and use up the fetal calf serum nutrients, they will change it yellow, and you pour it off again. And you can put them under a tissue culture microscope and look at them under like 10x and see these little things that look like neurons almost. They look kind of like this. And they just continue to divide until they cover the entire bottom of the surface of this flask. And you have grown up a complete monolayer. Well, that's not enough to get a production of virus, so you probably want to get more monolayer. So what do? We add more of this enzyme, trypsin. And the trypsin will release the monolayer. We shake it, add trypsin to it, release the monolayer, and pour it into 3 flasks. Lay them down flat so the human cells attach. Add fetal calf serum, and grow more monolayer. Then, once we get enough, we add the virus, which will infect, kill the cells, and they will release. And then the particles will be in the fluid. And we can harvest the virus from the fluid, from the fetal calf serum after. So we are growing on plastic flask monolayers of human or plant cells. How do we spread them? In plants, you would add just, you wouldn't add the fetal calf serum, you just add the nutrients, phosphate buffer, antibiotics, and and vitamins. And in human cells, you'd add the fetal calf serum, and when it turns yellow, you change it. And when it spreads and covers the whole bottom, you look through it through a tissue scope, and the whole bottom of the flask is one cell touching another one. You pour off the liquid, leave a tiny bit at the bottom, add trypsin, that releases the cell, shake it, pour it to like 4 or 5, lay them on their side, they attach, turn them back up, add more fetal calf serum, get more and more monolayer. Then you add the virus infects the cell, kills the cell, releases virus particles, and when they release, they're dead. So you harvest the liquid. So that's how we grow viruses. And this process is called cell culture or tissue culture. Cell culture or tissue culture. I usually had a picture. Hold on. Let's see if I've got it on my note. Yeah, there it is. So here we have all we did was take a closet. Went and rented a room in a regular old building, take a closet, put a heater in there on a thermostat, set it at human body temperature, build us some shelves, and then we put these plastic bottles on them. And we're growing monolayers, and then we infect them with virus, and nobody knows that right next to their post office or right next to their doctor's office, we're growing horrible human viruses in a closet next door. And we did it right near Santa Anita racetrack, 3 blocks away. We grew HIV and leukemia. One of the interesting thing is, if you want to grow a lot of cell culture at once, we have a cell hotel. It's about the size of my briefcase and about this tall, and there are 5 floors of cells.
How many diamonds?
So we have, what?
How many diamonds?
I don't know. We have a cell hotel, and it has these little drains, so if you pour the fluid in up here, it slowly rolls down here like this, and it keeps it all, and then you have the fluid drain out there. So and you can grow a lot of cells in a cell hotel. So everybody knows what a cell hotel is now? It's basically a whole bunch of plastic flasks stacked up together on top of each other, so you can grow a whole bunch of monolayer at once.
And the fluid that you're putting through, is that like serum, it's a
Yeah.
nutrient?
Yep. And we change it every time it turns yellow. You don't even have to say, oh my goodness, I wonder if they're hungry in 3 days. Nope, they'll turn yellow when they produce enough acid. All right, so where do we get our cell cultures from? This is kind of important. Where do we get them from? We can chunk a whole of herb or take a piece of some plant, and that's called primary cell culture, but it has a huge problem. Did you know that every cell in your body has a watch in it? Yep, every cell in your body has a watch called a biological clock. And every cell in your body has this thing that says, click, I'm old, die. These primary cell culture cells only live and reproduce a certain number of times. And it can be as many as 5 or as few as 3. So remember, the big problem with growing cell culture from a primary source, are you going to get lots and lots of monolayer? No, it's only going to divide 3 to 5 times. So can you get a lot of virus production? So remember, we do not use primary cell culture sources most of the time. Now, if I do take a big old chunk of it and put it in there to grow, one cell will overwhelm the rest, and that will be called a strain. So one of the cells I chunked out of her will overwhelm all the rest, and it will be the one that divides, and they will all be genetically identical. That's the test question. Which of our cell culture sources is genetically identical? And it is cells taken from primary sources, from an adult or a young animal or plant. What's the disadvantage of it? They only divide 3 to 5 times, so you can't get much. All right, so where do we get our cell lines that we produce all these vaccines from? Things like that? Believe it or not, we get it from diploid fibroblast cultures. And the one we use the most is fetal foreskin. Yeah. When they come in and say, oh, listen, you're going to have a baby boy, and listen, you really should have that baby circumcised. It's cleaner, it's less disease transmission because there's a 25% less risk if you're circumcised to pick up HIV than if you're uncircumcised. And it's traditional in this country, and other kids will make fun of him in gym class, and you just really should do it. Everybody does it. And I'll even give you a discount off your insurance. Instead of $1,500 for doing that little snip, I'll only charge you $8. And most people say yes. Guess what? They sell it for over $2,000. And they sell it for culture, because that's where we grow most of our vaccine cells. Why do we want to use fetal tissues? Because fetal tissues have no, remember, normal tissues, when they touch each other, they stop growing. Fetal tissues have no biological clock, and they have no contact inhibition. So the more you feed them, the more of them there are. So that's why they're wonderful for vaccine production. You don't have to worry about them only dividing 3 to 5 times. They will divide forever as long as you want. All right, and that's called a continuous cell line. Don't forget that. They are continuous. The other kind of cell line is cancer. And this is what we use really most of the time. You know, it's kind of hard to find all those fetal foreskins and all that. It's expensive and all. So if we want to get fetal collagen or fetal dermis cells, it's expensive. But it's not expensive to get cancer tumors. And what's good about cancer? Not much I can think of, but one good thing. They grow as long as you feed them, they will grow. They will pile up on top of each other. That's what a tumor is. But remember, these are not genetically identical, but since we're making viruses, we don't care. Remember, cancer is a mutation. So remember, they will have abnormal chromosome numbers and broken chromosomes, but we don't care, because all we want to do is make more viruses. What does the virus do? Takes over the cell, uses all the cell's energy to make viral particles, and then kills the cell when they're released. So remember, most research is done with cancer tissues and tumors. And they are immortal, continuous, and they're heteroploid. So tell me what heteroploid means. Not genetically identical. They have more than they have more or less numbers of chromosomes. Their chromosomes are not regular. All right, so anybody ever heard of HeLa? Ellen? Yes, this person had a cervical tumor, and they removed it, and it became the most widely used human cell line in the world. Every virus lab has HeLa cells. So this woman will be immortal forever. She now weighs hundreds of thousands of tons more than she ever did alive with cancer. She's the first immortal human. Of course, all it is a cervical cancer cell of hers, but anyway, there's some really good books written about this cell line HeLa. All right, moving boringly onward. What is meant, this is vocabulary, what's meant by a teratogen? A teratogen is a virus that results in birth defects. And so whenever you find out you're pregnant and you go to the doctor for the first time, they're going to take a blood test to see if you have any active infections with teratogens. Because if you do, they want to give you genetic counseling, because they can tell you how likely it is for your kid to have birth defects, and they'll probably recommend amniocentesis later on to see if there has been birth defects. So the name of the blood test that looks for teratogenic viruses is called the TORCH series. Okay, more vocabulary. CPE. Cytopathic effect. What is cytopathic effect? Well, it's a discovery that every time a virus infects a host cell, it will leave a visible change behind. You can't see viruses, but you can see CPE. And CPE is diagnostic. So many times, for instance, when you go and get a pap smear, by the way, don't listen to what Bush and all those people said about if you have 5 good years of pap smears, you don't need to do a pap smear but once every 3 years. Bull. Why? Yeah, statistically that's true, but that assumes that you get a good lab that reads your pap smear right. And I used to work in one of these medical technology laboratories, and let me tell you, there's some corruption. A lot of them are owned by doctors, and a lot of them are only there for the profit. And the company that I worked for was not owned by a doctor, it was owned by the mob from Delaware. Yes. And their job was just to squeeze anybody else out of the market and make as much profit as possible. So they underbid every medical laboratory in LA County for pap smears. And so they would get thousands every day. Except we only had 50 technicians to read them. And if they couldn't read them all in one day, and remember they go bad, they couldn't read them all, they would report them out as indeterminate. Take another sample. It didn't mean that they couldn't read the sample, it meant they didn't read it. But they charged for it now twice. And a lot of women go and then don't think about it, and if they do get a call back, your pap smear was indeterminate, it's not a crisis, nothing, no big deal, and they either forget about it or whatever. And if they went to that same place twice, they may not get red in several years. And then you have reading it wrong. My sister and my mother got breast cancer in the same year. My mother when she was 84 and my sister when she was 51. They do have the breast cancer gene, and in both cases it was hormonal because my mother took the hormone replacement for, what do they call it, menopause, and my sister took a birth control patch that was hormone based. And they both got breast cancer, yet every year they went for mammograms. And so after they both found the lump, my sister first and my mother second, my sister had a radical double mastectomy and my mother had a single mastectomy. And we went back and we said, where did you have your mammograms? And so we went to the hospital where they had their mammograms for 25 years and said, we'd like to see those mammograms. And they couldn't find them because they ran so many that the technicians either didn't look at them or read them wrong, and to keep from being sued, they lost them whenever somebody came back to check. So you want to get these things every year because there can be problems. Yes, statistically, you can do a study and saying if you've got a good smear and a good technician and a good lab, and you have 5 good years, then you can skip 2 years. That's true, but that assumes all of those things that you cannot assume. Well, anyway, what is CPE? Well, there's one virus that infects cervical tissue, a herpes virus. It infects the genital region, first herpes simplex 2. And if you take a scraping around the cervix and you look at this under a tissue culture microscope, the epithelial cells will seem to bubble. It will like have little bubbles projecting out of them, like that. And that is CPE for herpes simplex 2. That means that without seeing the virus at all, because the virus itself causes a change in the host cell that's visible under a simple microscope, tissue culture microscope, you can diagnose the illness with pretty good reliability, like in the 85%. And so CPE for different diseases is quite diagnostic. That's what a pap smear is. They're looking for changes in normal cervical cells. If the cells have fused together, in other words, several cells have fused together, and it is a giant cell with more than one nucleus, that is an indication that it's been infected by HPV, human papillomavirus, which causes cervical cancer. So a pap smear can tell you 2 things. Everybody knows what a pap smear is, right? Where they take just a gentle scraping around the cervix. And by the way, my niece, 27 years old, and refuses to go for pap smear. Makes me so angry because listen, there's lots of things they do to guys that are just as embarrassing. Like sticking their finger up your butt and grabbing your nuts and saying cough. And all these other things. And they do pap smears of men too, don't kid yourself, because human papillomavirus can be spread orally, it can be spread by all kinds of sexual, and it's the leading cause of neck cancer, anal cancer, and penile cancer. And you don't have to have sex in those areas to have the virus cause cancer in those areas. You just have to have transmission, and usually it's kissing and doing things with your mouth. Alright, so anyway, pap smear can diagnose 2 illnesses. It can diagnose a herpes infection. What's the CPE for herpes? Cells that seem to bubble. And what it can also to human papillomavirus. And what's it? Giant multinucleate cells. So these are both CPE that are diagnostic for virus infections. Another one is, how do we get a confirmed diagnosis of rabies? If somebody gets bitten by an animal that is rabid, they will look at the animal and see if it's acting weird. And if it does act strangely, like froth at the mouth and have a fever and be uncoordinated, they will sacrifice the animal. That means kill it, chop its heads off, and pack it on dry ice and send it to a lab. And in the lab, they will stain the neural tissue. And if it has dots in it, in the cytoplasm, those are dots of accumulated rabies virus. And those dots are called black bodies or negri bodies, and that is diagnostic for rabies. So that's still how they confirm rabies diagnosis, by doing stains of neural tissue. What about HIV? Well, HIV, this is what a T cell looks like. Within 12 to 18 hours of getting infected, this is what an HIV T cell looks like. This giant balloon will soon explode and release millions of virus particles, and that's called syncytia.
You can't remove it.
No. It's just hundreds of T cells get infected.
Plus, I mean.
So remember, what is CPE? A visible change in a host cell indicating a specific virus has infected it. What is CPE for HIV? Syncytia formation, this huge balloon. What is it for herpes? Cells that seem to bubble, epithelial cells that seem to bubble and froth up. What is it for HPV? Giant multinucleate cells. And of course, polio. What's the number one thing about polio? Atrophy. The cells don't seem to mature. Their maturity is stopped. So remember, CPE is very important, easy way to diagnose something that is incredibly small, difficult to diagnose, a virus. What do? You look at the host and see if there's been a change. Cytopathic effect. So the viruses we're going to concentrate on in the rest of this lecture is, we're going to go through the herpes. We're going to go through human papillomavirus that causes cervical cancer and genital warts. I'd like for you to look over polio, because what's this class about? Me, and I had polio. So be nice if you looked at it. And polio is going to be the second disease we hope to eliminate from the earth. What was the first one? One disease that we have eliminated, doesn't exist anymore except in labs. You don't get the vaccination anymore.
Smallpox?
Smallpox, correct. Smallpox has been eliminated from the earth except in the Soviet Union and the USA, where they keep it in a lab to weaponize it. And you remember Saddam Hussein bought it, and we don't know where he put it all. Hepatitis A, B, C, D, E, G. There is no F. The hantavirus pulmonary syndrome, I've almost told you everything about it. You need to know. Influenza, why we're so afraid about it. Leukemia viruses, HTLV1 and HTLV2. And human immunodeficiency virus, HIV/AIDS. So those are of the gazillion millions of viruses we're just going to talk about. And we're going to start with your friend and mine, things we love to share, herpes. Oh yes, there's nothing like a little herpes among friends. Well, first of all, herpes is a double-stranded DNA virus, and it's enveloped. So does that mean it's easy to kill in the environment or hard to kill? Envelope. Easy to kill in the environment, so people should not be giving it around. But because it's contact transmitted, meaning particles can be released by the hundreds of thousands from a lesion that somebody has, and it can get into any roughed up spot on your skin, People get it very easily. And on the tabletop, we can kill it. Easy. Disinfection, alcohol, detergents, all those things will kill it in the environment. Once it gets into a person, that's a totally different story. So remember, when I say easy to kill, I don't mean inside a person easy to kill. I mean that it is easy to kill on a surface. There's one. Let me get something real quick here. Hold on for 1 second. It's a simple virus. It only has 8 genes. And those genes, how they're arranged, determines which disease type of virus it is. It's a very strange one in that respect. In the fact that the same genes arranged in a different order give you a completely different disease. They're all herpes. So how do we call it? We arrange all the herpes by the genetic arrangement of the 8 genes. And so if it's arranged alpha style, these are called the dermatrophic viruses, the skin. And then if it's beta, those we call the teratogenic herpes. And then there's gamma. And the gamma arrangement is the cancer herpes. So we have herpes for everything. And by the way, let me just tell you, one of the exciting things about herpes is almost every animal has a herpes virus. And you do not want to get a herpes from another animal because you have no protection against it. As a result, it'll just run right all over your body. Plus, all of us have been exposed to a human herpes for quite some time. As a result, you have some defenses. In fact, most of you, if you haven't gotten the herpes simplex 1, the what we used to call oral herpes, if you haven't gotten it by the time you're an adult, you probably won't get it until you get old. Now when you get old, your immune system is kind of worn out, and you'll probably pick it up from somebody that has it. So let me just see if I can find the herpes video that's going to reinforce what I am telling you. And I'll be right back with you in just about 2 or 3 minutes here. You'd think it would just pop up, wouldn't you?
Our test bed? Yeah. Oh yeah, that's us.
There you are.
I got it.
Sad about herpes. All right. So anyway, let's continue with herpes. First of all, how would you be able to tell if you had herpes? What would they do? Pap smear or cell culture? That's the best way. You're going to be asked, what's the best way to diagnose herpes? Cell culture. That's what a pap smear is. You know, they're going to take, well, it's not what a pap smear is. What they do is they take a sample of your cells and put it on a slide and look for CPE. The best way to do it, though, is to take a sample and put it in a monolayer. Hey! Oh, thank you. Oh, thank you, Dick. No. Thank you. So anyway, the best way to detect a particular virus, particularly herpes, is to take a sample of the fluid from a lesion and send it to a lab, and they put it in tissue culture and look on a tissue culture scope, which shows a lot more cells than just scraping it, which is a pap smear. But so if I ask you which is the best way to diagnose herpes, you're going to say cell culture. What's the second best? Pap smear. Or a slight smear. Anyway, alpha herpes, there are 3.
Naked?
Nope.
No.
Naked viruses don't have an envelope. They don't have any, all they have is a nucleocapsid.
A capsid. Okay.
That's it, just a protein coat, but it's pretty indestructible. So here's some of the questions I'm going to ask you. Where does the envelope come from on an envelope virus? The plasma membrane of the host cell, it buds through it. And what does that envelope do for the virus? Allows it to attach to the same kind of host cell. And if you destroy the envelope, what happens to that virus? It's destroyed as well. All right? What about a virus that doesn't have an envelope? What's it called? Naked. And naked viruses, are they easily destroyed?
No.
No. How about enveloped?
Yes.
Name some things that will destroy enveloped viruses.
Alcohol.
Detergents, alcohol, disinfectants, antiseptics, phenol, all of those things like Lysol, anything with a detergent in it. Now remember, there's a difference between a detergent and a soap. All a soap is an alkali plus an emulsifying agent. So you have a very high pH, that's what alkali have, and what it does is it lifts oil and the dirt dissolved in it off your hands, and physically, with the foam, washes it down the drain. A detergent does the same thing, plus it inactivates enveloped viruses and bacteria. But most people have watched that television program that's been on since 1940, that television commercial where Madge is soaking in palmolive. And they said, oh, she's going for her manicure, and she says, oh, my hands have been sitting there. 5 for every 10 miles, 10 square miles. How often would they come across mouse pee? Hardly ever. Then we moved in, and we planted these lovely, lush lawns that we irrigate, which produce seed, which is what mice eat. And they move in, and they eat, and they live with us, and they live in our garages. And then on the weekends, we say, oh my God, the garage is just such a mess. So all the kids and I are going to get out there, and we're going to move everything out of the garage, and we're going to dust it, and then we're going to move it all back in and organize it. And you breathe the dust. And by the afternoon, by the evening, you have trouble breathing. Your lungs are filling up with fluid. They take you to the hospital. The hospital doesn't think about this disease because it's used to be so rare. And remember, your doctors are trained, what, 10, 15 years ago. This disease wasn't even seen until 1997. So they don't think about it because it has all the symptoms of viral pneumonia. But it progresses so fast that you will be dead by day after tomorrow. Now what they're supposed to do is put you on a respirator and give you steroids to cut off your immune system. But that's not what they normally will do because they haven't isolated and identified it, and they don't want to cut off your immune system if it's a bacterial or another kind. So what happens is the virus that you breathe in lodges in the cells of your alveoli, like the little capillaries lining the alveoli that absorb oxygen. In your bloodstream are what we call cytotoxic T cells, killer T cells. And these killer T cells will attack any host cell that's infected with a virus. Now, if your killer C cells don't attack, people live there. 5 per every 10 square miles. How many people live there now? 500 per square mile. So we had a disease that didn't exist, or we didn't know about it. It existed for 5,000 years that we know of because the Navajo talk about it. And it's called, now we call it Southwestern Pulmonary Ponton, which I never spell right, I hope it's right, virus syndrome. And it's carried by the California deer mouse. The normal brown mouse that you see running around here is a deer mouse. It has ears that look just like a deer. That's why they call it the deer mouse. And it has this virus living inside of it happily. They coexist. They love each other. They're symbiotic. The virus doesn't hurt the mouse, and the mouse doesn't attack the virus. They live together in harmony and love. And whenever the mouse poops or pees, they pee out, just like we poop or pee out anything we're infected with. They poop or pee out this virus. But when humans breathe dust that this virus has peed on, that these mice have peed on, you get southwestern pulmonary hantavirus syndrome, and it will kill you in 4 days.
Wow.
And it kills you by your own immune system killing you. You kill yourself. Your immune system kills you. So here's what happens. We didn't see that virus because people live a rough, and just, yes, you should change to a better dishwashing detergent. Something like Palmolive that's kind to your hands and your skin. And she said, well, I'm worried about using something like that. And she said, well, you're soaking in it. And she goes, oh! And then says, well, it's fine because it's okay. Well, remember, detergents have a reputation for drying and making chapped, cracked hands, which they do. But soaps don't kill chemically, only physically. So you want, but could Dial sell Dial antibacterial detergent to you to wash your hands with? You wouldn't do it. But what did they change it to? Dial antibacterial soap. Is it really a soap? No, it's a detergent, but they couldn't market it as a detergent because people thought, oh, it'll dry out and crack my hands. So they sell it as Dial antibacterial soap, but it's not a soap. Now, one other thing, remember, anybody, most of you women in here have knowledge about this, most of the guys don't, but I'm sure you know that if you're taking care of your beautiful hair, you don't use Pert. Does anyone know what Pert is? Pert is this product that they sell that says it is a shampoo and a conditioner in one. You can shampoo and condition at the same time. You cannot. Conditioners are oils. Shampoos are detergents. What do detergents do? They remove oils. So you cannot add an oil to a detergent. If you do, you've done nothing. So when you go and buy something to wash your hands with, I wish we used to have some here, I guess we don't. You want to buy a antibacterial soap, but you don't want to buy one containing lanolin or skin softeners because it's an oil. Oils hide bacteria and dirt. So it's sort of like this. You can't jump on top of the car and go to school. You got to get in, you got to go through the process. And so you wash your hands very well with a detergent, and then you go get a good hand cream to moisturize them and keep them from cracking. You don't try to do it all at once. I know they try to sell you the product, but it won't work. In fact, it's more hurtful because it traps dirt and bacteria in the oil droplets. So remember, you've got to wash first, then moisturize. Never try to do it at once. So emerging viruses are viruses we've never seen before because we didn't live there. Do you know what they used to call what's that area around San Bernardino called what do they call that? The something inland empire. You know, they called along here the Gold Coast. They call that the Inland Empire, which is marketed. But what did we used to call the Inland Empire 20 years ago. Desert. But you don't want to say, let's move to the desert. Move to the Inland Empire. Now, so what was the Inland Empire? Desert. How many people?